So it's a really great pleasure to welcome Camille Truong, who's giving a talk here from Melbourne. You're in Melbourne again now after being in Tasmania, aren't you? Is that right?
Yes, yes. I came back on Monday.
Good, okay. And she really kindly agreed to give this talk at reasonably short notice, so I am really grateful. Camille did her PhD at the University of Geneva in Switzerland, and she's done research in temperate and tropical forests of the Americas, Africa and Australasia. And she is now working at the Royal Botanic Gardens in Victoria. So welcome to Australasia. And yeah, it's really great to be able to meet you and to hear about some of the research you've done. So I'll just hand it over to you and you can share your screen and hopefully it'll all work fine.
Well, thank you so much, David, first of all, for the invitation. And this has been a really great symposium so far. So today, I'm going to talk a little bit about ectomycorrhizal fungi and the role that they have for the nitrogen economy of nothofagus. We're going to cross continent here and talk a little bit about nothofagus that are actually in Patagonia, which is where I've been doing some work before coming here. So I'm sure you all know this famous scene from The Lord of the Rings, where the hobbits, they walk in the forest and they stay for the night, and then they suddenly wake up during the night and they realize that the trees are talking to each other. And so even though we know that the trees, they don't use words like in the movie, they are able to communicate between each other using what we call the mycorrhizal network. So it's a network of fungi that will connect the roots of one tree to the other. So it comes mycorrhizal, it comes from Greek, that's a fungus associated to roots. And so these fungi, they receive carbon from the tree, from the photosynthesis, and in exchange, they will give to the tree nutrients and also some resistance to pathogens and hydric stress, for example. What is important to remember about mycorrhizal fungi is that it's actually a highly heterogeneous group. So there are many different kinds of mycorrhizal fungi, and even though they all trade nutrient against carbon with the plants, they do it in very different ways. So it's actually difficult to make general assumptions about mycorrhizal fungi because they have very different strategies to get nutrients and give it to the plant. And even within the group of interest that I'm going to talk today, the ectomycorrhizal fungi, they have different origins, and so they also have different ways of doing this. It's a little bit, if you want, like both bats and birds do fly, but the evolutionary history that they have to be able to do this is actually quite different. So this is important to remember this. In the case of ectomycorrhizal fungi, something that is also important for ecosystems is that in general, in these forests, more than 50% of the carbon that is fixed by photosynthesis is stored below ground. So it transferred below ground to the ectomycorrhizal network. So we're talking about a huge amount of carbon. And this is really important in terms of carbon cycling, or for example, climate change, how to fix the carbon from the atmosphere. So it's a really large amount of carbon that is stored below ground. And so if we look a little bit more into plant nutrition, so the plants, they get their carbon, of course, from photosynthesis, but they also need to get minerals such as nitrogen, phosphorus, or potassium, other things. And so the plants, in order to absorb these nutrients, it needs to absorb it in an inorganic form. So, for example, when a deciduous plant loses its leaves in the fall, and the leaves fall onto the ground, the plant doesn't have access to the nitrogen that is in the dead leaves. This nitrogen needs to be first mineralized, and this is done by microorganisms such as bacteria and fungi. And it's only once the nitrogen is mineralized that the plant can absorb it through the roots. But some groups of ectomycorrhizal fungi, for example, the genus Cortinarius, which is highly abundant in nothofagus forest, can access organic forms of nitrogen in the organic matter. What does it mean is that they have enzymes to degrade organic matter, get the nitrogen and get it to the plant directly without having to do mineralization. So it's some kind of shortcuts. The fungi, the plants doesn't need to have mineralization anymore. It can get the nitrogen directly from the organic matter through this group of ECM fungi. Not all ECM fungi do this, but Cortinarius is an example of this. And so it's important to remember that even though these ectomycorrhizal fungi, they can degrade organic matter, they're not saprophytic. Because if you see on this scheme here, they still get their, they still get their carbon from the plant. This is like the black arrow here. So they get the nitrogen from the organic matter, but they get carbon from the plants. While the saprophytic fungi, they get both their nitrogen and carbon from the organic matter. So do these ectomycorrhizal fungi degrade organic matter? Yes, they have enzymes to do this, but they're not saprophytic. They also have slightly different ways of degrading the organic matter to access this nutrient. They mostly do it through oxidation. So they have oxidative enzymes to do it, such as peroxidases, for example. While saprotrophs in general, they use hydrolytic enzymes to degrade organic matter. So they also do it in different ways. But because of these abilities, there have been some models that have been developed about ectomycorrhizal forest. Which is, they call it the ecological market or the nitrogen economy of these forests. So basically what it means is how do the plants trade the nitrogen with the mycorrhizal fungi compared to what is the stock in the soil. So it's really kind of a market thing, right? And basically, what does it mean? What these people showed is that when we have reduced nitrogen availability in the soil, so there is reduced inorganic nitrogen available to the plant directly in the soil, the plant is going to increase the carbon that it gives to the ectomycorrhizal fungi in order to get nitrogen directly from these, right? And because these ectomycorrhizal fungi, they don't have to go through this mineralization, this will increase the immobilization of the nitrogen, so the nitrogen will be even reduced in the soil, and etc., etc., etc. And this is what we call about, the forests are trapped in nitrogen limitation. And this is one explanation why a lot of temperate and boreal forests, the nitrogen is heavily limited. But even though, despite this, we still have this huge tree that have a very high demand of nitrogen, but the nitrogen available in the soil is very limited. So this is a well-known model that many people have been studied, but most of the studies have been done in the Northern Hemisphere, especially in the boreal forests that are dominated by conifers, especially the genus Pinus, so the real pines. And so we were interested to see what is happening in temperate forests in the Southern Hemisphere that are actually dominated by ectomycorrhizal plants that are angiosperms. And so in order to do this, we established our study model in Patagonian forest. This is usually when I give talks in the Northern Hemisphere. This is when I spend a lot of time talking about the fact that there are actually temperate forests in the Southern Hemisphere. But you guys know all this, so I will be really short here. I will only point out a few differences between Patagonia and Australasian forests. The first one is that in these forests, the dominant trees are broadleaf notophagaceae. So they form ectomycorrhizal association. And so in Patagonia, they are the dominant forest trees. There is no other ECM families in this forest. So there's no Myrtaceae, no Rhamnaceae. This is the only ECM host are notophagaceae. There are, of course, exotic pines and eucalypts, but in the native forest, they are the only trees, and they are dominant. There are some conifers there, such as here, Podocarpus, Fitzroya, Pil, Gerodendron, but they're never dominant. They're usually emergence tree, and they also associate with different types of fungi, which are arbuscular mycorrhizal association. So there is a very interesting biogeographical pattern in Patagonia where the notophagaceae, they've been isolated. They have not been in contact with other ectomycorrhizal hosts for a very long time. And the other difference I'd like to point out is that in the south of Patagonia, in subantarctic forest, deciduous tree species dominate. So we have a landscape that we see, like you can see on the photos, which is something that we would never found in Australasia because a lot of the nothofagus species are actually evergreen. And so if you think in terms of carbon and nitrogen cycling, this is a huge input of nitrogens that comes every fall in the soil, in the leaf litter, and which obviously have a lot of impact on nitrogen cycling and the ectomycorrhizal fungi that we're going to find there. And so the research question that we had was to understand the role of ECM fungi for the nitrogen economy of nothofagus in Patagonia. And we had the following hypothesis. We think that the nitrogen availability will correlate with the nutrient demand of the different nothofagus species that we found there. And then secondly, we think that the dependence on the ECM symbiosis will increase with nitrogen limitation, similarly to what has been shown in coniferous boreal forests. And so in order to test this hypothesis, we established our study plot in the archipelago of Tierra del Fuego. So if you can see on this map, we're here at the southern tip of South America. And we chose this place because it has very strong climatic and edaphic gradients. So we are here at the tip of South America. The Andes mountain range which is running from north to south. Here runs from west to east, like this. And so there is a huge gradient in precipitation. The southern part of the archipelago gets a lot of rain. We're talking about 2,000 to 4,000 millimeters every year. And then we have this huge rain shadow by the Andes Cordillera. And on the other side of the mountains, the rain drop conspicuously. It's also much more continental. And actually, you can see that on the northern part of the archipelago, it's actually a steppe vegetation. There's not even forest anymore. So on a few hundred kilometers, you have this huge gradient. It was also interesting for us to do a study there because there are 3 nothofagus species that grow in Tierra del Fuego: Nothofagus betuloides, Pumilo, and Antarctica. And as you can see on this map, even though they enter in contact with each other, they are heavily structured and distributed according to climate, water regime, and soil properties. So they really form this monospecific forest with a single ECM host that sometimes enter in the contact zones, but the mixed forest is actually very narrow there. So they have very contrasting ecologies, the 3 of them. And so here today, I'm going to concentrate on these 2 species, Nothofagus betuloides and Nothofagus pumilo. So Nothofagus betuloides is what we call the beach comber because this is the species that go along the coast and major lakes with the highest rainfall. It's also an evergreen species. It grows on highly acidic soil. We're talking about pH 3 or even less than this. With a very thin organic horizon, you see sometimes it almost grows on the rocks. And these forests, they're also seasonally waterlogged. And based on previous study, we know that they have a poor nutrient content of the leaves. So poor nutrient content, especially of nitrogen and phosphorus. And so the assumptions that we always had about this is that they grow on nutrient poor soils. And the other species, Nothofagus pumilo, is what we call the climbers because it grows along the mountain slope in the interior. So it may seem to be weird, but it's actually the same species that grow all the way from the valley up to the tree line. On these photos, you only see one species, Nothofagus pumilo. It's a deciduous species. This is the species that you show on the photos at the beginning. It grows on soils that are better drained, and it also higher nutrient contents of the leaves. So the assumption that we had was that it also grows on more fertile soils. So what did we do here? We sampled soil from Nothofagus betuloides forest, Pumilio forest, and also in the mixed transition zones. So we had a total of 90 samples. And with this sample, for each one of them, we identified fungal communities using the ITIS-1 barcodes on the Illumina MySeq platform. We also did enzymatic assays. So we measured the activity of fungal enzymes in the soil, 8 of them. And then we also measured edaphic variables such as the pH, soil moisture, the carbon-nitrogen ratio, the inorganic nitrogen and phosphorus, so available to the plants. And then we did two things with the nitrogen. We measured the net nitrogen mineralization after 30 days incubation. So basically, you take your soil sample, you incubate, you measure how much nitrogen available you have, and then you incubate it for 30 days and see how much nitrogen was mineralized by the microorganism during that time. And then we also measure the microbial biomass of nitrogen, which is you do a lysis of the microorganism in your soil sample, and so you see what is the nitrogen that is actually not released in the soil, but inside the body of the microorganisms. And so we did all kinds of modeling with this data. We looked at fungal community structures, differences between the two species, the mixed forest and the pure forest. But I promised David I would be good and not go over 30 minutes. So today I will focus really on the nitrogen cycling. And so we have these two species, Betuloides, which is evergreen and has a low nitrogen and phosphorus content. And then we have Pumilo, which is deciduous and has a high nitrogen and phosphorus content. But when we look at nitrogen mineralization in the soil, we actually found that there were more inorganic nitrogens in the Betuloides forest, and also more mineralization, more potential for mineralization in the Betuloides forest, which is somehow contradicting our hypothesis of that Betuloides was growing in less fertile soil, because there's more nitrogen available in the Betuloides forest, even though they apparently need less nitrogens than Nothofagus pumilo. So it was a little bit surprising at first. I remember that with the student that was doing this analysis, we actually asked her to redo them and check, because we were a little bit surprised at first. But then after analyzing the data again, we realized that it actually makes a lot of sense. Because what we also found is that especially this is one example of the enzymes that we studied. So this is a fungal enzyme that is responsible for getting nitrogen out of the organic matter, so leucine aminopeptidase, was actually much higher in Nothofagus pumilo. And also the microbial biomass N, so the nitrogen that is inside the body of the microbes. So we could not decipher between bacteria and fungi, but this is a lot of them. The nitrogen inside the microorganism was much higher inside the pumilo forest. And so in terms of nitrogen market, we think this is what is happening is basically in Nothofagus betuloides. It's an evergreen, so we have relatively little fall in the soil, and then relatively little nitrogen is going back to the tree because they have low nitrogen and phosphorus requirement. As I was telling you, there is higher inorganic nitrogen and mineralization in the soil. We also have a low soil pH and a relatively high CN ratio. So basically, this means that there is not a lot of nitrogen in the soil organic matter. But because there is low microbial nitrogen and the low activity of the nitrogen acquiring enzyme leucine aminopeptidase, we are in what we call typically an inorganic nitrogen economy. So there's not a lot of nitrogen available. And because remember, there is this waterlogging from time to time. This is what we call an accelerated nitrogen economy. So basically it's not a lot of nitrogens, but it's very rapidly recycled. So rapid mineralization and the nitrogen get in the soil, get mineralized and back to the tree very rapidly. While on the other hand, when we look at Nothofagus pumilo, we have the exact opposite. The tree is deciduous and has a high nitrogen and phosphorus content, so it has somehow high demand of nitrogens, but there is very low inorganic nitrogen and mineralization in the soil. The soil pH is also relatively higher, and the CN ratio is a little bit lower. So it means that there's more nitrogen trapped in the organic matter, but it's not mineralized. So how do the tree get this nitrogen? This is what we call an organic nitrogen economy. So basically all the nitrogen that we found in the soil, most of it was within the body of the microorganism. And we also find this high activity of the fungal enzyme leucine aminopeptidase. And so basically, this is what we call this organic nitrogen economy that is really regulated by ECM fungi. So basically, there's ECM fungi, such as the genus Cortinarius that I was telling you about. They are able to get the nitrogen directly from this organic matter and give it to the tree. And so even though there is very low inorganic nitrogen and mineralization, the tree still get the nitrogen it needs through this ECM fungal network. And so this is typically what has been described as an organic nitrogen economy previously in the Northern Hemisphere in coniferous boreal forests. Oh, my screen is blocked. Okay. And so when we looked a little bit at the major ECM fungal families that we found in the different types of forests, this correlated really well with our assumptions, because we found that fungi from the family Clavulinaceae, they were the most abundant species in terms of both species diversity and also frequency in the sample. And these fungi, they are heavily adapted to inorganic nitrogen uptake, while, like I was telling you in the introduction, species from the genus, the family Cortinarius, sorry, but it's mostly the genus Cortinarius. They are heavily adapted to organic nitrogen uptake, and so they were much more abundant in the primordial forest. And this was interesting because we really showed that these forests, they are functionally very similar to coniferous boreal forest, even though we are talking about angiosperms and we are in the southern hemisphere. And so in conclusion, what we found is that we did not find the nitrogen availability correlated with the nutrient demand of the different notophagous species. We actually found exactly the opposite. But because of this organic nitrogen economy, we could show that the dependence of the ECM symbiosis seemed to increase with nitrogen limitation, similarly to what was found in coniferous boreal forest. And so one question always leads to another question. We now are exploring if this is actually favoring the monodominance of Nothofagus pumilo, because in this forest, Nothofagus pumilo is the only ECM host, but there's also very few other plants, even though they are associated with other types of mycorrhizal fungi, both in the tree layer or in the understory. And so imagine that if the nitrogen is so heavily limited in this forest, if you're not an ECM plant, you don't have any nitrogen available to grow on. And that's why we think that potentially the plant diversity in this forest is so low. And so before I take your question, I just would like to mention about this mycorrhizal network that we have established in South America, especially if there are students listening to this talk today who would like to connect with a student from Chile or Patagonia working on similar things. So basically, with this network, we would like to promote research and outreach in South America and beyond. We have students from all over the world now. We like to have a very horizontal interaction. So it doesn't matter if you're a student or if you're a professor, everyone has a voice and everyone can share information. So basically, we organize a symposium. The next one is in Colombia. It was supposed to be 2022. It's going to be 2023 in August next year. We also have a Facebook page where you can connect, diffuse information. We also do interviews about scientific articles. So if you have a publication that you would like to diffuse more widely, we can make a little interview to you, and they are actually broadcasted on the newsletter of the International Mycorrhizal Association. So it's a good way to reach out to many people. And with this, I would like to thank my sources of funding, the agencies of Switzerland, USA, and also Argentina, and all my collaborators, which, as you can see, a lot of students were very keen of doing soil sampling in very harsh conditions, and I'm happy to take your questions. Thank you.
Great. Wow. Thank you very much. What a fantastic talk. Okay, we do have a few questions. Teresa, I think I've just made you a co-host. Are you in a position to ask your own question? Do you want to do that?
Sure. Well, it's not really a question. It's more of a comment about the eucalypts particularly in southern Western Australia, do leaf drop, and it can be quite a significant amount of leaf litter that pulses into these systems. And yeah, I think it would be fascinating to look at some of these kind of, and I have a feeling they'll probably end up being close to that rapid mineralization economy kind of model.
Yeah, I agree. This is a very good point. I feel that the main difference with this deciduous forest is the time scale, because they basically they lose all their leaves within a very short period of time. The summers are really short in Patagonia. The snow often comes really early. So there is this like 2 weeks period where these enormous forests are going to lose the leaves. So the input in the soil is like really, really big. And I'm not sure that that would be the same for species that, of course, the evergreen, they also lose the leaves, but it's kind of more in a like extended ways. But yeah, this is definitely something that we're looking into it. And I was in Tasmania recently looking at this population of the Nothofagus gunnii, who is this like the remnant deciduous forest in Australia. And so I think this, yeah, I have to say that, so there's nitrogen economies, they're not necessarily linked to a species in particular. It's more linked to ecological conditions. So depending on the condition, the same species could have different nitrogen economies. But this is, yeah, definitely something that has been studied so much in boreal forest, and we still know so little about what is happening here. So I think that's very interesting.
Great. Yep, I was going to ask that about the leaf fall too, because I think, yeah, it's probably not as quick as 2 weeks, but I've heard people say, you know, that it's 14 months sort of leaf life cycle, that sort of thing. But okay, Peter J, you're unmuted, so you can talk, and then while you do that, I'm going to get David, you're online if you want to.
Yeah, just a quick question. I might have missed this earlier in the talk, but in your chart of the families detected, they were all basidiomycetes. Did you? I presume you found some ascos on the roots as well.
Yeah,
did any
actually,
of those maybe have a mycorrhizal lifestyle as well?
Yes, that's a very good point. Actually, we did find several ascomycetes that are ECM, especially in the Pedzizales order. So the genus Notojaphnia in the Tuberaceae, Aluhaina in the Pyranomataceae. And so these fungi, so far, what we found is that they are very abundant with seedlings. And also they seem to have really big impact for like forest regeneration. For example, in this case, the graphics I showed you were from like the mature forest. So we didn't find they have so much role, but they have a very important role for seedling recruitment, for example. So yes, we did find some, and I'm actually really interested to look also at just the diversity and taxonomy of these. So thank you for asking.
Great. Okay, David, you should be able to unmute yourself too. Oh, there we are.
Right. Yeah, I'm still trying to wrap my head around the northern hemisphere coniferous forest, where you obviously don't have leaf litter, but are the I guess is the litter from the pines, is that enough to give the quaternarias there? Or is there quaternarias on coniferous forest? That's maybe the first question, and then secondly.
Yeah, so in the boreal forest, Cortinarius was really the model that they have been used to show this organic nitrogen economy. But I think it's also because we know from their genomes that they have these peroxidases enzymes to do the degradation of the organic matter. That's actually a good question about the leaf litter, because I mean, in boreal forest, there is often quite a lot of pine needles on the soil, but yeah, I agree, they are not deciduous, obviously, and they can be very recalcitrant, so, you know. So there are obviously differences in the fact that this is a coniferous and here we have angiosperms, but I thought it was interesting to see that there's actually different, very similar patterns in the way the nitrogen is recycled in like the two types of forests, and because it's a similar ectomycorrhizal fungi that do this, even though they associate with very different types of trees. But yeah, that's a good point, actually.
Okay, yeah, that makes sense, I guess, because it's the evergreens on the northern hemisphere that do that, but not the evergreens in the southern hemisphere. So it's really interesting.
Right, thank you.
Thank you.
All right. It looks like that's about all of our questions, and we've just got out of time. So thank you so much, Camille. It's fantastic.